Hello everyone, and welcome to today's presentation. I'm excited to have you here as we explore the fascinating world of DNA. The title of today's talk is Structure of DNA, with a focus on understanding its function and characteristics. Over the next few slides, we'll dive into what makes DNA so essential to life, how it's built, and what unique features it has. Let's get started. Today, we'll be outlining what we'll cover in this lesson. First, we'll look into the characteristics of DNA, understanding what makes this molecule unique and essential. Then, we'll explore the function of DNA, which is at the heart of how living organisms develop and operate. After that, we'll delve into the discovery of DNA, looking at how scientists uncovered its structure and importance. These points are all connected because understanding DNA's characteristics and functions helps us appreciate how it was discovered and why it is so fundamental to biology. By the end of this lesson, you'll have a clear picture of DNA's role in life itself. Let's take a closer look at how DNA fingerprinting plays a crucial role in forensic science. While it doesn't give us a definitive answer about who committed the crime, it significantly narrows down the list of suspects. Here, we see a crime scene sample and the DNA profiles of Suspect 1, Suspect 2, and Suspect 3. Each suspect's DNA has specific regions called loci, which are like unique markers. In this example, we have 3 loci, locus 1, locus 2, and locus 3. By comparing these loci between the crime scene sample and each suspect, we can see which profile matches most closely. The goal is to identify which suspect's DNA closely aligns with the DNA found at the scene. This comparison helps investigators focus their attention more effectively, bringing us closer to solving the case. Remember, DNA fingerprinting is a powerful tool, but works best when combined with other evidence to build a strong case. Today, we'll be focusing on some key learning outcomes that are essential for understanding DNA. First, we'll explain the primary function of DNA, which is to store and transmit genetic information. This is what makes it the blueprint of life, guiding all biological processes. Next, we'll look at the characteristics of DNA, such as its double helix structure, its stability, and how it encodes information through sequences of nucleotides. Understanding these characteristics helps us appreciate how DNA can reliably carry genetic instructions across generations. Finally, we'll identify the scientists who uncovered DNA's structure, notably Watson and Crick, whose discovery was groundbreaking in genetics. Connecting these points to the overall theme, recognizing DNA's function and structure is fundamental to understanding how life is inherited and how genetic information influences living organisms. Let's talk about some of the key figures behind the discovery of DNA's structure. Rosalind Franklin and Maurice Wilkins played crucial roles in this scientific breakthrough. Franklin, in particular, used X-ray crystallography to capture images of DNA, which revealed important details about its form. Maurice Wilkins collaborated with her, and together they provided vital data. On the other side, James Watson and Francis Crick took these insights and built on them. Using the data from Franklin's X-ray images, they were able to develop a model of DNA's structure. Their work culminated in a groundbreaking conclusion in 1953. DNA is shaped like a 3-dimensional double helix. This discovery was a huge milestone because understanding DNA's structure is fundamental to grasping how genetic information is stored and transmitted. It's a perfect example of how collaboration and different scientific techniques, like X-ray crystallography, can lead to major breakthroughs. This discovery not only advanced biology, but also set the stage for countless future research in genetics. Let's take a closer look at the DNA double helix. As you can see, DNA, or deoxyribonucleic acid, is made up of 2 nucleotide chains that are twisted into this iconic double helix structure. Each of these nucleotide chains is composed of smaller building blocks called nucleotides. Now, each nucleotide itself is made up of 3 parts: a sugar molecule, a phosphate group, and a nitrogen-containing base. This combination is what gives DNA its unique properties and ability to store genetic information. It's important to remember that DNA is essentially a long chain of these nucleotide monomers linked together. Understanding these characteristics helps us appreciate how genetic information is organized and passed on. So, in summary, DNA's structure and its components are fundamental to its role in genetics and heredity within all living organisms. Let's take a moment to explore this short segment of the 3D DNA molecule. Feel free to click and drag to rotate it around and get a better view. As you look, see if you can identify the sugar phosphate backbone. That's the structural frame of the DNA. Also, look for the nitrogenous bases, which are the rungs of the ladder. Now, why is the twisted ladder shape so important? Well, this helical structure helps protect the genetic information inside and allows the DNA to be compacted efficiently within the cell nucleus. It also plays a key role during replication, making it easier for enzymes to unzip and copy the genetic code. Understanding this structure helps us see how DNA is not just a static molecule, but a dynamic, carefully organized blueprint of life. So, take a moment to explore, and then we'll discuss your observations and thoughts. So, here we see a segment of DNA, specifically the sequence CGCT88ATGCG, along with its complementary strand, CGC88ATGCG. This shows just a short stretch, only 12 nucleotide pairs, highlighting a small part of the much larger DNA molecule. Now, the reason DNA looks like a twisted ladder, or what we call a double helix, is crucial. This structure not only makes it compact and easy to fit inside the cell, but also provides stability. It ensures that genetic information is stored securely and can be accurately copied when cells divide. Understanding this structure is key to grasping how genetic information is maintained and passed on, which ties into the larger theme of how DNA functions as the blueprint of life. All right, let's take a moment to check our understanding with this quick knowledge check. As you can see, this slide encourages you to customize the poll or quiz by double-clicking or visiting the Elements tab for more options. The key here is engagement, making sure you actively participate and apply what we've discussed. Remember, these interactive elements are designed to reinforce your learning and keep you involved. So take this opportunity to reflect on the main concepts we've covered so far, and don't hesitate to experiment with the poll or quiz tools to deepen your understanding. Connecting this to our overall theme, staying engaged and interactive helps solidify your grasp of the material and makes the learning process more effective. Now let's take a closer look at the base pairs in DNA. Cytosine, which we abbreviate as C, always bonds with guanine, G similarly, adenine, A pairs with thymine, T. These specific pairings are crucial because they ensure the stability and integrity of the DNA structure. The hydrogen bonds between these complementary bases act like glue, holding the two strands of DNA together. This bonding is what keeps the double helix intact and allows DNA to carry genetic information accurately. Understanding these base pairings helps us appreciate how genetic information is stored and transmitted from one generation to the next, which ties into the broader theme of DNA's role in life itself. All right, let's take a moment for a quick knowledge check. As you can see, this slide is all about engaging you with a poll or a quiz. You can easily customize it by double-clicking on the element or heading over to the Elements tab for more options. The key here is to make sure we're actively testing your understanding of the material. Remember, these tools are designed to reinforce learning and keep you engaged. So don't hesitate to participate. It's a great way to connect the dots and deepen your grasp of the overall theme we're exploring today. Did you know that the sequence or order of the bases in a specific DNA strand actually plays a crucial role in determining our traits and qualities? It's really fascinating to think about how just the arrangement of these tiny building blocks can influence who we are. For example, if we look at a DNA segment with the sequence GTCATT, that might be responsible for traits like curly brown hair. On the other hand, a very similar sequence, GTCAAT, could lead to straight black hair. So even a small change in the sequence can make a big difference in our physical characteristics. This highlights how the genetic code is essentially a set of instructions that dictate our unique features. It ties into the overall theme of how genetics influence our appearance and traits, showing just how powerful and precise these sequences are in shaping us. Now let's talk about the function of DNA. Essentially, DNA acts as the blueprint for life. It stores all the genetic information that organisms need to develop, survive, and reproduce. Think of it as the instruction manual that guides every biological process. But it's important to remember that this information isn't static. It's delicate and can be damaged. When DNA gets damaged, it can have serious consequences. For example, cells might stop dividing, which affects growth and repair. In some cases, damage can lead to cell death, and in more severe cases, it can cause cells to become malignant, leading to cancer. So, understanding the function of DNA and the importance of maintaining its integrity is crucial, because it directly impacts health and the proper functioning of living organisms. This highlights how vital DNA is not just as a genetic storage system, but also as a key player in health and disease. Let's move on to our knowledge check. This is a great opportunity to review what we've covered so far. I want you to think about the key points we've discussed, especially how they connect to the overall theme of our presentation. Remember, the goal here is to reinforce your understanding and clarify any questions you might have. So, take a moment, and let's see how well these concepts are sinking in. Feel free to ask questions or share your thoughts as we go along. This is all about engaging and ensuring we're on the same page. Now let's explore how these structures fit together. On this slide, I want you to interact with the 3D model and locate key components: DNA, histone, nucleosome, chromatin, and chromosome. As you do that, keep in mind that if you uncoiled the DNA in each cell, it would stretch out to about 2 meters. That's quite astonishing considering how tiny the cell nucleus is. This incredible length of DNA needs to be packaged efficiently to fit inside the nucleus. So, it first wraps around histone proteins, forming what we call nucleosomes. Think of these as the beads on a string. These nucleosomes then coil further into chromatin fibers, which are more compact. Finally, these fibers condense into the visible structures we refer to as chromosomes. Understanding this process, from DNA to chromosomes, is crucial because it shows how genetic information is organized and stored. It's a beautiful example of nature's efficiency, ensuring that all our genetic material is safely packed yet accessible when needed. So, as you move through the model, try to see these layers of organization and appreciate how they relate to the overall theme of genetic regulation and cellular function. Now let's talk about DNA function. DNA is essentially the blueprint for life, as it stores all the genetic information an organism needs to develop, grow, and reproduce. Without this information, life as we know it wouldn't be possible. Moving on to the characteristics of DNA, it's fascinating to see how it's made up of nucleotide monomers. These are the building blocks that form the entire molecule. There are 4 key bases in DNA: adenosine, thymine, cytosine, and guanine. These bases pair in specific ways and encode the genetic instructions. The discovery of DNA's structure was a major milestone. The double helical model we refer to today was the result of collaborative work by scientists like Watson, Crick, Franklin, and Wilkins. Franklin's X-ray diffraction images were crucial in revealing the helical structure, which Watson and Crick then modeled, transforming our understanding of genetics. To sum up, DNA's ability to store and transmit genetic information is central to biology. Its molecular structure, composed of nucleotides and characterized by the double helix, underpins all of life's complexity. This discovery not only explained how genetic information is organized, but also opened the door to countless advances in science and medicine.